Hello, and welcome to my channel. My name is Greg Friedman, and I'm a plasma engineer focused on development and research in electric discharges. Now, on this channel, we chat about the papers that I find interesting. Looks like most of you aren't subscribed according to YouTube statistics, so please subscribe. Click the little bell icon on the bottom, and I would love to have you as a regular. Please let me know what you think of this channel in the comments. It's brand new. There are only 300 subscribers. This is video number 15. So I would love some feedback, maybe papers you'd like to learn more about. But anyway, let's get to today's paper. So I'm looking at this paper that recently came out in 2021, and it's an optimization of application of plasma functionalized water for microbial safety and fresh cut endive processing. I had to look up what endives are. Apparently I've been eating them, just never knew they're called endive, and they're very awesome, very tasty. Highly recommend them. And they're also pretty high value products. So application of new technology, such as plasma functionalized water, is interesting for this new type of an application. So let's dive right into it. Of course, these researchers are talking about disinfection, and they compare this new method with the currently industry accepted method of using chlorine dioxide. Introduction chats about lettuce and other fresh fruits that are minimally processed because they can get damaged if you start tumbling them or use any harsh chemistry. Many of us know that Listeria, Salmonella, E. coli are occasionally detected in fresh produce and occasionally kill people. So it's nice to get rid of them. In my best tradition, I keep finding little mistakes. So this is O157H7 E. coli, and here is O158H7. It's just a mistype, but it's funny that peer-reviewed, published manuscript with a bunch of editors still gets little mistakes like this. During the fresh produce washing, we can introduce cross-contamination because we wash one dirty leaf and it can contaminate other leaves. So it's important to look at removal of E. coli both from the surface of the fresh produce, but also from water. And, these researchers talk about the other water additives that include chemical sanitizers like ozone, hydrogen peroxide, electrolyzed water, peracetic acid, or physical treatments such as, high pressure, pulsed electric field, oscillating magnetic field, ultraviolet, gamma, etc. This plasma treatment is sort of between the two. It's a bit of chemistry and a bit of physical, and it's a very interesting technique. As the authors demonstrate, it's quite cost effective, but we will hear about that later. The authors reference you to previous work where they fully characterize the system, and we don't really need the details, not for this brief review. They're using this Blesk, whoever came up with that name, but it's a two-stage microwave plasma running at 2.5 gigahertz. Looks like that. It's very fancy. Microwaves are pretty hard to work with, and these guys are treating 60 liters at 4.3 kilowatt. So they're using 72 liters per minute of air, running it through 4.3 kilowatt plasma torch, and then bubbling it through water, I guess. That wasn't fully mentioned, but then I didn't read the original work. So that's 4.3 kilowatt for 1 hour. That's 4.3 kilowatt hours. That's pennies. So this is under $1 for 60 liters, bringing it to cents per liter, which is already pretty reasonable. They are able to treat up to 20 liters simultaneously. So this is a bench scale system, yes, but already, getting into gallons per hour, which is quite impressive. The authors attribute disinfection to runs, reactive oxygen and nitrogen species, and acidity, and they provide all of the measurements. So a lot of the details I'm brushing over. All the links to the paper are down in the description. Please feel free to make your own judgments. I'm just giving you a short overview of items that I found interesting here. Here are some properties of the plasma processed air and plasma functionalized water. This should be H2O2. All these little mistakes, come on. Maybe I'm a stickler, but I don't let my students get away with this. But anyway, I keep reminding myself that PPA stands for plasma processed air, and PFW stands for plasma functionalized water. And get this, PTW is undefined, but I think it's plasma treated water, which now they're calling it plasma functionalized water. Just these little oversights. It's okay. So anyway, the important thing, they do antimicrobial assay, very standard, and they calculate reduction factor. So basically this is comparing how many bacteria there were before the treatment versus how many there are, and taking log of that. So if we had 1,000 and now we have 10, that means we got 100-time reduction or a 2-log reduction, otherwise known as 99%. They also measure metabolic activity. So we're looking at how many bacteria will grow on a petri dish. They're also looking at metabolic activity, if the bacteria that are on the produce are breathing. They're also measuring live dead, so vitality assay. So this is a pretty strong paper doing significant amount of work to analyze the results they're getting. They go into lots of different processing. So when they take feedstock, they slice off the outer leaves, they pre-bathe, they pre-rinse, they pre-wash, the main wash and post-rinsing. So there are 1, 2, 3, 4, 5 stages to the wash. And then they put this produce without tumbling it, just let it drip off a little bit. And there are lots of details how the measurements are done. I don't think they're essential for this conversation. If we look at the antimicrobial efficiency data, or I should say when we look at the antimicrobial efficiency data, we're comparing plasma treated water, plasma functionalized water with tap water. So we're comparing dunking this lettuce or whatever it's called, endive, dunking these endives into tap water compared with dunking them into plasma functionalized water. And take a look here. So we're comparing red, which gives us 5 log reduction, with tap water, which gives us barely 1 log. So out of A, B, C, D, E, we really should look at D, which is the best for plasma, with E, which is chlorine dioxide, industry standard. And chlorine dioxide gives us a log, if that, depending on where the fresh produce is extracted, while plasma treated water in this particular setting gives us 5 logs. All I take away from these graphs is there exists a setting on this machine that gives us a 5 log reduction compared to 1 log reduction of industry standard. So this is pretty great. And if we look at metabolic activity, again that point D shows that plasma treated water, metabolic activity is virtually 0. So whatever's there isn't breathing much. Well, compared to tap water, whatever's there is nearly at 100%. That's good news. Let's compare it to chlorine dioxide. Well, that's cool. So tap water, same as with plasma treated water, or with control in the plasma treated water, whatever's there is breathing. Here, whatever's there is breathing. And chlorine dioxide, same thing. So whatever there is pretty alive. I found that interesting. However, when we look at microbial vitality, the assay that looks at live dead, we see pretty much no difference. So whatever bacteria are collected are stained live. Interesting. It's an observation. While they don't breathe and they don't grow, the ones that are stained with live dead assay look to be alive. Okay, interesting. It's it's a relatively short paper with some very interesting results. Chemical analysis shows that nitrites and nitrates are the predominant species in this water. And this is typical for microwave discharges. They're hotter discharges, so it's going to be mostly reactive nitrogen species. They do measure a tiny bit of peroxide, about 80 millimolar, which is way below, if you take 3% hydrogen peroxide, that will be about 900 millimolar. So we're significantly below something that's typically used for disinfection. However, I've seen lower numbers, especially in acidified environments. Well, it is what it is. So they do measure a little bit of it. And there's a discussion about the potential mechanism for gram-negative versus gram-positive. It's interesting details, but I didn't find them interesting enough to share with you. Feel free to download this paper and go into details yourself. Of course, the authors mentioned the peroxynitrate, which is the hot molecule these days. Well, anyway, so the results we observed are pretty excellent. However, the authors immediately go into criticizing themselves for the scalability. This is a bench-top type of experiments. They're treating 60 liters per hour, getting to pH below 2 at a cost of 1.5 cents per liter. So application to 400 kilograms of lettuce seems possible. Although, of course, there are many challenges during scale-up. So in conclusion, what we've observed here is there is a novel system giving you up to a 5 log reduction on the surface of fresh cut produce when this plasma activated water, plasma treated water, plasma functionalized water, whatever you want to call it, when that's used for fresh produce washing. I think this technology is excellent. Use of microwave discharges is a little bit unnerving. They're usually finicky, require high purity gases. So I wonder what the system would behave like in environment of production plant at Yuma, Arizona. But still, this is quite promising. I would be very interested at looking into different discharges, like my favorite gliding arcs would work really well for this. And they also are hot discharges or transitional discharges, so there would be a lot of reactive oxygen, reactive nitrogen species. Bottom line of this is, looks like plasma activated water is entering as a contender in fresh produce disinfection, and I am very curious to see more of these publications and commercial implementation of this. Again, please subscribe, click the little bell icon. I would love to hear your feedback, what you want to see more of, what you want to see less of, and look forward to making more of these videos for you. Peace, guys.